This video is an introduction to neuropathology, detailing the structural and functional organization of the nervous system. It explains the embryogenesis of the nervous system, from the neural tube into the different segments of the brain. Terminologies related to structural anomalies, motor disorders, metabolic diseases, and inflammatory conditions affecting the nervous system are also covered.
All right, welcome. Today we are diving deep into the architecture of you. We're going to explore the absolutely incredible way our nervous system gets built from the ground up. And then we'll look at what happens when that master blueprint has an error, or when the finished structure starts to break down. So let's jump right in at the very beginning.
You know, it all boils down to this one question. How is this even possible? It sounds kind of crazy, right? That this unbelievably intricate network that controls every single thought, every feeling, every movement, starts out as just a humble, flat sheet of cells. That transformation? Well, that's the heart of our story today.
So how does an incredible construction project like this even get started? Well, like any great building, it all begins with the architectural plans. We've got to go way back to the very beginning, to the early embryo, where the foundation for the entire nervous system is first laid down. And the single most important material for that foundation? It's a layer of cells called the ectoderm. Seriously, think of this as the raw lumber, the steel, the concrete, everything you need to build the skyscraper that is the nervous system. From the brain all the way down to the tiniest nerve ending, it all originates right here.
Okay, what happens next is this kind of mind-blowing cellular origami. It's just amazing. A little section of that ectoderm thickens up, and then its edges, the neural folds, they start to rise up, almost like you're folding a piece of paper in half. They come up, they meet at the top, and then they fuse together, creating this hollow, closed structure, the neural tube. This little tube, this is the precursor to the entire brain and spinal cord. And get this, every single one of these steps has to be absolutely perfect.
All right, so with the main structure framed out, the system now needs its specialized workers, right? It needs its materials to fill in the walls. So let's zoom in to the cellular level and see just how this blueprint gets populated. You see, the walls of that neural tube we just saw are just packed with these immature cells that are going to become the specialized team for the central nervous system. You've got your neurons, they're the messengers, the signal transmitters. Then you have the glial cells, your astrocytes, oligodendrocytes, and ependymal cells. They're the crucial support staff doing all the maintenance. And then you have the microglia, which are so cool because they're basically outsiders. They actually migrate in from a totally different embryonic layer to act as the resident security and cleanup crew of the CNS. Now, as all these cells get organized, we form two really distinct types of tissue. And honestly, the easiest way to think about it is this. Gray matter is like your computer's processors. It's where all the neuronal cell bodies hang out and do the actual thinking and computing. White matter, on the other hand, well, that's all the wiring. It's just bundles of long, insulated cables, we call them axons, that transmit the signals between all the different processors and out to the rest of the body. And to really wrap your head around the complexity of that gray matter processor, you have to understand the neuropil. It's not just a bunch of cell bodies sitting in neat rows. Oh no. It's this incredibly dense, tangled-up mesh of all their connections, the axons, the dendrites, and all the arms of those support cells. This feltwork, as it's called, is where the real magic happens. This is the physical stuff of communication in the brain. So here's the crucial point. This whole construction process is exquisitely precise. And because it's so complex, with so many steps that have to happen in a perfect sequence, it's also incredibly vulnerable. So now we're going to shift gears and look at what happens when there are errors in that initial blueprint. Remember that amazing folding process we just saw, where the neural folds fuse together to create that perfect tube? Well, the technical term for when that seam doesn't close up properly is dysraphism. It literally just means abnormal seam. It's such a simple name for a problem with such profound consequences. And the consequences of this faulty closure? They depend entirely on where along that seam the failure happens. As you can see here, if it happens down along the spinal cord, it can lead to spina bifida. If the seam fails to close at the head, it could be cranium bifidum. And in the most severe cases, like anencephaly, a major failure of that seam at the head means that large parts of the brain just don't form at all.
Another major problem that can happen during development is hydrocephalus. The hollow inside of that neural tube we talked about eventually becomes the brain's ventricular system, and it's filled with something called cerebrospinal fluid, or CSF. If the drainage system for this fluid gets blocked for any reason, the fluid backs up, it accumulates, and it creates this intense pressure that can seriously damage brain tissue.
Okay, so far we've been focused on errors in the initial construction. But what happens when the system is fully built, everything's looking good, and then things start to break down later in life? Now we're going to shift our focus to diseases that affect the system's components and its day-to-day functions.
To do that, first we need to speak the language. I know, neuropathology terms can look super intimidating, but they're usually just logical descriptions built from smaller, simpler pieces. Let's try one out.
Okay, get a load of this word, polioencephalomalacia. That's a mouthful, right? But believe it or not, it's actually a very precise description of a specific problem. It's not as scary as it looks.
Let's just break it down. Polio, that refers to gray matter. Encephalo, that just means brain. And malacia is a term that means softening, which in pathology usually means tissue is dying. So, put it all together, you get softening of the gray matter of the brain. See? It's just a code, and once you know the pieces, you can crack it. Pretty neat.
All right, let's talk about another really key process, demyelination. Remember our analogy of white matter being the electrical wiring of the nervous system? Well, myelin is the rubber insulation on those wires. Demyelination is exactly what it sounds like. It's when that insulation gets destroyed, causing the signals to slow down, get scrambled, or just stop altogether.
And this failure of the insulation, well, it's at the heart of a lot of different disorders. You can kind of group them into a few buckets. Some, like degenerative myelopathy, are about the progressive breakdown of myelin that used to be perfectly healthy. Others are congenital, like hypomyelinogenesis, where the myelin just fails to form properly right from the start. And then you have others, like Krabs disease, that are caused by metabolic problems that actively destroy the myelin.
Now, a really common result of all these different breakdowns is something called ataxia. And this is so important to understand. Ataxia is not a disease itself. It's a clinical sign. It's a clue. It's that loss of coordination, that wobbly, clumsy movement that tells the doctor, hey, something is wrong in the parts of the nervous system that control movement, especially in a part of the brain called the cerebellum.
And that one sign, ataxia, can point to a whole bunch of different root causes. It could be from a cerebellum that just didn't develop properly, which we call cerebellar hypoplasia. It could be from the premature death of neurons in the cerebellum, a condition called an abiotrophy. Or the problem might not even be in the brain at all. It could come from something like Wobbler syndrome, where the spinal cord is actually being compressed up in the neck. So after looking at how this incredible system is built in all the ways it can break down, what's the big picture here? Why does understanding this intricate blueprint really truly matter? At the end of the day, the key takeaway is this. The very same complexity that makes the nervous system so unbelievably powerful also makes it incredibly fragile. Every single step, every cellular migration, every connection, it all has to happen just right. This incredible intricacy is its genius, but it's also its Achilles heel.
This quote from our source material just sums it up perfectly. It's not a random, chaotic process. It's a series of coordinated steps. Think about it like a symphony orchestra. Every single musician has to play their part at exactly the right moment for the masterpiece to emerge. Just one wrong note can change the entire composition.
And that leaves us with this final, crucial question. As we learn more about the astonishing precision that's required to build this system, from the folding of a single sheet of cells to the wiring of trillions of connections, it really forces us to ask, what more can we do to protect this fragile masterpiece, both as it's developing and throughout our entire lives? Thanks for joining us.